Good morning. It's January 2nd, and this is your daily brief in science. Here's everything you need to know. A new practical framework aims to reshape how clinicians identify nociplastic pain in routine visits. Duradia has introduced the Duradia Protocol, a 10-point checklist designed to help recognize mixed pain mechanisms rather than labeling pain as purely nociplastic or not. The approach emphasizes that most patients exhibit mixed mechanisms and should avoid black and white classifications. The protocol focuses on strategies to modulate the nervous system and improve functional outcomes while avoiding unnecessary procedures. Once nociplastic pain is identified, the treatment plan shifts away from repeated procedures toward calming and retraining the nervous system, organized around 3 pillars: explain, move, and calm. Explain is patient education, move covers graded activity to rebuild function, and calm addresses sleep, mood, and stress management. Education, graded activity, and lifestyle factors become central to care, reframing treatment around these 3 pillars. The goal is to calm and retrain the nervous system through this framework and reduce reliance on invasive tests when central sensitization is the dominant mechanism. The article notes that educating patients reduces fear, which can lower pain intensity and boost engagement in recovery, a point highlighted by Dr. Gautam Das. Understanding nociplastic pain is presented as a real, actionable diagnosis intended to prevent unnecessary tests and treatments by correctly identifying the dominant pain mechanism. The piece includes contact details for Duradia and standard legal disclaimers and is provided as is by EIN Presswire with typical accuracy caveats. A high-fat diet can push liver cells into a survival mode that primes the organ for cancer, and in studies, nearly all mice on such a diet developed liver tumors, while humans with liver disease showed similar pro-survival gene patterns linked to poorer outcomes after tumor development. Over roughly 2 decades in humans, cancer risk accumulates, but the timeline varies with alcohol use and overall health. Repeated metabolic stress from excess fat intake activates survival pathways in hepatocytes and suppresses normal metabolic and protein secretion functions, a trade-off that keeps cells alive but raises tumorigenesis risk. Some signaling pathways tied to this reprogramming may already be druggable, with SOX4 activation highlighted as a potential indicator of misregulation and a therapeutic target. Researchers are testing strategies to reverse the reprogramming and to develop drugs targeting the molecular switches, with findings available online in Cell. Experts are exploring whether dietary changes or GLP-1 weight loss medications could reverse or mitigate the cellular damage. Current work points to possible interventions including metabolic drugs and liver-stabilizing therapies, but it remains unclear if the cellular state can be fully reversed through diet or medications. SOX4 is normally active mainly during fetal development and is rare in healthy adult liver tissue, making it a notable research target. Practical implications include early risk stratification using gene and protein signatures, new targets for prevention and treatment, and considering diet or GLP-1-based therapies to counter stress responses. Future work will determine whether returning to a healthier lifestyle can reverse cellular changes and halt cancer progression, including testing diet, weight loss meds, or targeted therapies. Researchers will test if reversing high-fat-driven changes through diet, weight loss medications, or targeted therapies can restore normal liver cell behavior and prevent cancer progression. Transcription factors in genes regulating this immature state shift, such as thyroid hormone receptor, HMGCS2, and SOX4, could become drug targets to reduce cancer risk in at-risk individuals. A single 10-minute burst of intense exercise may trigger molecular changes in the bloodstream that could slow bowel cancer growth and boost DNA repair. In a Newcastle University study, blood samples taken before and after a short cycling session were used to expose bowel cancer cells in the lab, revealing changes in gene activity linked to metabolism and DNA repair. Researchers found about 13 proteins rose in the blood after exercise, with many associated with reduced inflammation, better vascular function, and altered metabolism, suggesting exercise signals can influence cancer cell behavior. Gene activity shifts included upregulation of mitochondrial energy metabolism genes and downregulation of genes tied to rapid cell growth, alongside activation of DNA repair through the PNKP gene. Bowel cancer is the fourth most common cancer in the United Kingdom, with about 44,000 new cases each year and a high mortality rate. Risk can be influenced by lifestyle. The study involved 30 overweight or obese healthy men, and a separate trial with 30 adults aged 50 to 78 showed the same short cycling bout raised at least 13 circulating proteins, including IL-6, and altered cancer cell gene expression when serum was applied to lab-grown bowel cancer cells. The study design focused on pre- and post-exercise blood samples to assess effects on cancer cells exposed to serum. Beyond exercise, the findings align with broader guidance that high-fiber foods from whole grains, pulses, fruits, and vegetables, while avoiding processed meat and limiting alcohol, can lower bowel cancer risk. Regular activity, including gym workouts, sports, active travel, and routine tasks like gardening or cleaning, remains part of a wider approach to reducing risk. A multimodal deep learning framework called ImmunoStruct is advancing how we predict immunogenicity by integrating diverse data types to forecast immune responses to neoantigens. It combines AI techniques with structural biology to identify immunogenic epitopes that can guide personalized therapies for infectious diseases and cancer. By tying immunology, structural biology, and AI together, the approach leverages predicted neoantigens to inform cancer immunotherapy strategies. Key data sources include IEDB and CEDAR for epitope data, with the MHC motif atlas providing binding specificities. Ongoing efforts aim to expand peptide MHC coverage and deepen collaboration between computational biologists and immunologists to translate predictions into vaccines and therapies. Validation shows SARSC-CoV-2 epitope predictions align with in vitro results, underscoring potential for rapid vaccine development against emerging pathogens. Foundational work spans TCR recognition, antigen processing, MHC presentation, and advances in neural networks and protein structure prediction relevant to immunology. Core methods blend graph neural networks, attention mechanisms, and self-supervised learning with structural biology of peptide MHC complexes and tools like AlphaFold, NetMHC-PAN, MHC-NUGGETS, and MHCF-LURRY. Clinical and translational angles are evident through phase trials and immunotherapy approaches such as neoantigen vaccines, TCR therapies, and checkpoint blockade combinations. Trained on a large multimodal dataset of over 26,000 peptide MHC interactions, ImmunoStruct learns beyond sequence data alone and aims to predict survival outcomes from these interactions to personalize therapies. Multimodal data integration yields a robust, nuanced view of epitope presentation and recognition, with implications for vaccine design and cancer treatment strategies. A massive Bronze Age settlement may redefine Ireland's prehistoric towns. New work at Brusselstown Ring in County Wicklow shows more than 600 suspected roundhouses clustered inside and around its inner and outer enclosures, suggesting a population hub on a hilltop dating from roughly 1293 to 410 BC. Radiocarbon dating and 4 test excavations indicate occupation from the late Bronze Age into the early Iron Age, with dense dwellings but no clear social hierarchy signals in the artifacts recovered so far. Airborne surveys reveal 98 house platforms inside the inner enclosure and 509 between the inner and outer ramparts, painting a picture of substantial density for a hilltop site in this region. Preliminary results show contemporaneous occupation across houses of varying sizes, with no direct link between house size and artifact richness, mirroring patterns seen at other Bronze Age domestic sites in Ireland. The broader abandonment of Brusselstown Hill in the Iron Age appears linked to regional population dynamics rather than Bronze Age climate shifts, with work led by Dr. Dirk Brandherm of Queen's University Belfast synthesizing 2 decades of surveys and 2024 excavations to map interior layout and enclosure features. Future investigations aim to confirm enclosing elements, refine chronology, and determine the cistern's function and significance, including dating the stone-lined, boat-shaped cistern near a trench that could be a Bronze Age water storage feature, the first of its kind found in Ireland if confirmed. Another trench area points to a Bronze and Iron Age water cistern capable of supporting a large population, underscoring the site's scale and its place in the Baltinglass Hillfort cluster, which dates to around 1200 BC and hints at proto-urban development earlier than is typical for Irish settlements with only a few dwellings. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. We'll be back Monday with everything you need to know.